Great. So we started our discussion of the electron transport chain and chemiosmosis, right? This last stage of oxidative phosphorylation of cellular respiration. So remember, the electron transport chain and this process of chemiosmosis both take place on the inside of the mitochondria, right? And right here in this image, we are looking at that inner membrane of the mitochondria, where you have all the various components of the electron transport chain embedded, along with the ATP synthase, or that enzyme that ends up making the ATP. Okay, so when the citric acid cycle is finished, and the majority of the energy from glucose still remains as those high energy electrons that are being stored by NADH and FADH2, both of those electron carriers are going to come to the electron transport chain within that mitochondrial inner membrane, and they're going to drop off those electrons, right? And the electrons are going to start here at complex 1 or at complex 2, depending if they're getting dropped off by NADH or FADH2, and they're going to move through the electron transport chain, much like you see here, that yellow arrow, and as they move through, remember, each of the proteins within the chain is playing hot potato. We're having redox reactions. One protein gets oxidized as another one gets reduced, and then that one gets oxidized and the next one gets reduced. And as these electrons are passed down, they give up some of their energy, and that energy will eventually be used to make ATP. Now, while this particular image shows you how those proteins are embedded in the membrane, it doesn't really give you a very good idea of how those electrons are losing their energy as they go from complex to complex. So this next slide shows you a graphical representation of the various components of the electron transport chain in sort of, again, a graphical fashion that will chart out the progress of the electrons through the electron transport chain in respect to the amount of energy that the electrons have. So if we take a look at this graph, on the y-axis here, you have free energy. So the higher up on the y-axis we go, the more free energy those electrons have. And then on the x-axis, down here, we have sort of progress through the electron transport chain. So you'll notice we'll start here with that first complex, second complex, third, and then fourth, and then all the way down here, remember, the last place where the electrons go is that oxygen, that final resting place for the electrons that started out in glucose and made their way all the way through cellular respiration, and they end up with that oxygen. Also, the way that those protein complexes are set up, remember, many of them are made out of multi-protein sort of subunits that are all built together to form that one larger complex. And so if you take a look, for example, 1 here has 2 of those, and then we move on to complex 3, we have 2 of the proteins out of the 3 as your cytochrome proteins. Cytochrome proteins, these are going to be important protein complexes because they are unique in that they have an extra iron atom in them. And much like, for example, hemoglobin has a heme group with an iron in the middle, and this is what helps the hemoglobin carry oxygen within your red blood cells. The cytochrome protein components of these electron transport chain complexes allow the electrons to be pulled down the chain towards the oxygen. All right, and so the proteins within these larger protein complexes, again, remember, will play hot potato with those electrons, and so they end up alternating oxidized and reduced states. So they become oxidized when they lose their electrons, and then they become reduced when they accept electrons from the protein complex further upstream. And most importantly, remember that the electrons get dropped off here at the very top of our graph, or the top of our electron transport chain. And as they move down, with each subsequent handoff, they give up some of their energy. So by the time that they end up at the very bottom and drop down here all the way to oxygen, they've exhausted essentially all the energy that they have to give. Oxygen will pick them up. Oxygen also will grab some of the protons or hydrogen ions from the solution to eventually form water, which is one of those products of cellular respiration. Okay, so at this point, if we think about it, glucose is done, right? We finished degrading that at the end of the citric acid cycle. And now, at the end of the electron transport chain, after that first half of oxidative phosphorylation, we're done with the electrons. But what are we missing? We still have yet to make those 28-ish ATPs that we were promised when the glucose molecule first went into cellular respiration. So if the electrons gave up all that energy, where did that energy go? So let's take a look. The next slide shows you that the electron transport chain itself produces no ATP, right? So on this right-hand side of this image, there is no ATP. But what there is, if you look very closely, this is something we haven't pointed out in these figures yet, as the electrons start here at NADH and FADH2, and they make their way through the electron transport chain, each time the electrons pass through complex 1, or complex 3, or complex 4, the energy that the electrons give up is used to pump protons or hydrogen ions across that membrane, across that inner mitochondrial membrane. And these protons or hydrogen ions are pumped from the matrix in here into that intermembrane space, that space in between the two mitochondrial membranes. And so these hydrogen ions, they just build up there. Right? This is an example of active transport. We're using energy, right? So energy comes from those redox reactions as the electrons go through the electron transport chain. And number two, we are actively pumping those hydrogen ions across the membrane against their concentration gradient, right? So those are the two, remember, qualifications for active transport. You're pumping something against its concentration gradient, and that requires energy. In this case, we've got both, right? We're using the energy from the electrons, they give it up, and we're pumping hydrogen ions across the membrane against the concentration gradient. And so what this does is it creates this high hydrogen ion concentration gradient in that intermembrane space. Those protons, those hydrogen ions, they don't want to be there, right? They want to go back to where their concentration is low. This is diffusion, right? That natural tendency to want to spread out to where the concentration is less, right? Moving down the concentration gradient. So naturally, all of those hydrogen ions are going to want to come back across the membrane. But you know, the mitochondria says, no, wait, I just expelled all this energy, passing all these electrons down to get you concentrated within that intermembrane space. You can't just come for free, all right? So what I'm going to do is I'm going to let you come across my inner membrane, but you have to do a little bit of work for me. And so the mitochondria allows those protons to come back. But when they come back, the only place they can come back into the matrix is through the ATP synthase. And again, this is the enzyme using that flow of protons back into the matrix, allows it to have enough energy to take ADP and inorganic phosphates and link them back together to form molecules of ATP. So now, through the use of the electrons falling down the electron transport chain, giving up their energy to pump those protons into the intermembrane space, as those protons flow back in, we use that energy much like a hydroelectric turbine. Instead of lighting a light bulb, we end up linking ADP plus inorganic phosphate to make ATP. Okay, so this slide essentially revisits the same thing which we just mentioned with the previous slide. So the electron transfer and the electron transport chain causes the proteins to pump those hydrogen ions from the matrix into the intermembrane space. Those protons, or hydrogen ions, move back across the membrane. The only place they can do this is through the ATP synthase. Right? And this ATP synthase then uses that exergonic flow of the protons back into the matrix to drive phosphorylation of ATP. All right? This is an example of a process that we call chemiosmosis, or the use of the energy that's stored in that high hydrogen ion gradient to actually drive cellular work. Right? So if we go to the next slide. Right, this is sort of a zoomed up view of that ATP synthase. Here's our intermembrane space where you have that high hydrogen ion concentration. Down here is the mitochondrial matrix. Here's where you have low hydrogen ion concentration. The hydrogen ions want to flow from the top here into the matrix. As they do this, so here's our little hydrogen ion, it's going to come in, and as it flows through, it's going to spin this rotor here, which spins the catalytic portion down here of that ATP synthase, and that will then give that enzyme enough energy to take ADP here and the inorganic phosphates and and link them back together to form ATP. This process of using that energy that's stored up here in that high hydrogen ion concentration is called chemiosmosis. It's used then to link ADP and inorganic phosphates to make ATPs. All right, so here, the energy stored in that proton gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis, puts those two parts of oxidative phosphorylation together. Right? That high hydrogen ion concentration gradient links them together. We have a very special name that we give to that high hydrogen ion gradient in that intermembrane space. We call this the proton motive force. And it emphasizes the capacity of all those hydrogen ions squished together in that tight space. It emphasizes that its capacity to do work, right? So we have two very similar words, right? On the previous slide, we have the word chemiosmosis. This is actually the action of using the proton motive force to do work, or the action of using that high hydrogen ion concentration to do work. That's chemiosmosis. Whereas the actual physical high concentration of hydrogen ions that has the potential to do work but may not yet actively be doing work, we call that the proton motive force. Right? So two very similar concepts there. Make sure they're pretty clear. At least differences between the two. Right? So chemiosmosis is more the action, and then the proton motive force is more of an adjective or a noun, for example. Okay, so we've gone through essentially all of cellular respiration. We've taken glucose, we've broken it down, we've extracted all of its energy, we've used that energy to make ATP. So if we were to review, if we were to do sort of an accounting of all the ATP that's produced by cellular respiration, we can essentially follow the flow of energy, first of all, right? So energy starts out at the very beginning in a molecule of glucose, right, our fuel source. That energy is extracted from glucose by the removal of those electrons. Those electrons are given to the electron carriers, NADH for example, and NADH hangs on to those electrons and shuttles them safely, because it's precious cargo, safely off to the electron transport chain. In the electron transport chain, as the electrons bounce down, their energy is passed now from the electrons, which give it up, to the active transport of those hydrogen ions into that intermembrane space. So we've moved the energy now from the electrons to the proton motive force, that high hydrogen ion concentration in that intermembrane space. And then we use the energy that's stored there in that proton motive force. As those hydrogen ions flow back across the membrane, we use that energy to make ATP, and the energy is then stored in ATP until the cell uses it to do whatever cellular work it needs to do. Alright, so now, if we think about it, cellular respiration, this is a really complicated process. The downside is that it's actually not very efficient. Only about 34% of the energy in a single glucose molecule is actually transferred to ATP during cellular respiration, and this ends up making about 32 ATPs, which we discussed earlier. Now, 34% doesn't seem very efficient, right? But if you think about it, your car, for example, might only be in the 20s in terms of efficiency, how much energy it can actually use from the gasoline in the tank to physically drive your car. And the rest of it is lost as heat, right? The engine gets really hot in a car. And the same thing happens for our cells and for our bodies, right? As we go through cellular respiration, we inadvertently produce that heat. This is what helps us maintain body temperature. Now, in the next video, I'll talk about accounting a little bit more. So why is it that we can't exactly pinpoint how many ATPs are produced from one glucose molecule?